Welcome, everyone, to the UCSD Osher Lifelong Learning Program. This morning will be our third speaker in our Master Class series of CARTA experts. CARTA is a Center for Academic Research and Training in Anthropogeny based here at UCSD. On October 24th, they presented their fall symposium on the human-ape paradox. If you took advantage of the chance to watch this free online, you had a very insightful and thought-provoking experience. You can check out their website at carter.anthropogeny.org if you missed the event. And the date for their upcoming winter symposium will soon be posted. It is titled Altered States of the Human Mind, Implications for Anthropogeny. And the co-chair for this event is our speaker today, Dr. Patricia Churchland. Dr. Churchland is Professor Emerita of Philosophy and former Chair of Philosophy at UC San Diego. She is also an adjunct professor at the Salk Institute, where she was strongly supported by Jonas Salk and Francis Crick. Born in Canada, Dr. Churchland has degrees from the University of British Columbia, Pittsburgh, and Oxford. She taught philosophy at the University of Manitoba before joining UCSD. In 1991, she was awarded the MacArthur Prize. She has written many books, one of which was the first to bring together computational concepts and behavioral data, all within a neurobiological framework. Titled The Computational Brain, it is a seminal textbook and recently had its 20th anniversary release by MIT. Today, she will delve into the social and neurobiological roots of human moral behavior as she speaks to the brains behind morality. Welcome, Pat.
Thank you so much, Eileen. And it's a great pleasure to be able to give this talk. And I love this collaboration between Osher and CARTA. I think it works really well. Now, I changed the title slightly to Social Conscience, but it's basically a story that I've talked a little bit about before to Osher, but there are also new data that I think it sort of embellish and enrich the basic story. So in 1975, the great biologist Ed Wilson said that the evolution of human sociality is the fundamental conundrum of biology. And it was an interesting thing for him to say because, of course, he had worked on social insects, in particular on ants. And he realized that there is a tremendous difference between the sociality of ants, which is really quite closely tied to the genes that make the wiring of their little brains. And in contrast, human sociality is very rich, very complex, and involves a huge amount of learning. Now, to start to think about where social values might come from, especially in the case of mammals, it's good to think about where any kind of value comes from. And the deepest level of value, of course, has to do with one's own welfare and survival. And this is true of all organisms. As Paul MacLean used to put it, you have to survive, you have to thrive in the 4 F's: feeding, fleeing, fighting, and reproduction. Failing that, you tend to become somebody else's dinner and you fail to pass on your genes. So the deepest level of value really does have to do with one's own survival and well-being. And understanding that, motivated a number of biologists, Richard Dawkins being one of them, to think that social behavior in humans, and in particular moral behavior, was not something that we were inclined to do for hereditary reasons, but rather we kind of had to have it beaten into us. And so he said, we are born selfish. Now, of course, that's partly true, but we are also born deeply social. And Darwin understood that. In 1871, in his book, The Descent of Man, he asks the question, where does our moral sense or conscience come from? And he proposed that there were three components: social instincts, whereby we are by nature inclined to be social. But the form of that sociality and the shape that it takes depends on the community into which we are born and the habits and the skills that we acquire. And finally, he thought that problem solving in a particular environmental context and social context might mean that the norms and habits that you learned as a child would change. Now, Darwin wasn't the first to propose this. Aristotle actually had a framework for thinking about morality that was very similar to that. And Confucius actually does too, and so do the two great Scotsmen, David Hume and Adam Smith. Now, what is new, I think, is the appreciation of the mechanisms whereby we acquire habits and skills that one might call social skills and moral skills, and why these kinds of social instincts developed, particularly in mammals and birds. Many sciences have contributed to this understanding, and in particular, I think that the ethologists who study animal behavior, both in the wild and in captivity, have been extremely important. Here, of course, we see a pair of chimpanzees, one consoling another after a defeat. But we also see pro-social choice, or, for example, in food sharing. Remarkably, in the wild, there are a number of instances of males who have adopted orphan chimp babies and have taken care of them and taught them how to crack nuts, let them ride on their backs until they were adults. We see other aspects of social behavior, such as cooperation, self-control, third-party punishment. It's not exactly the same as it is in humans, but we see instances of that kind. And it's not just restricted to primates. We see in the case of rats that one rat will overcome its fear of going into an open area to help to try to release another rat that it knows from a trap. So there is something very interesting and intriguing about the social behavior of mammals. In this slide, I just want to remind us that, of course, there are many species of hominins. On the vertical axis, you can see years in millions. Horizontally, you see geographical spread. Homo sapiens have been around not very long, about 300,000 years. Homo erectus, who had a much smaller brain, about 800 cc's, whereas yours is between 1,300 and 1,500. Homo erectus was around for about 1.8, 1.9 million years. They had controlled use of fire. They appeared to have lived in relatively small groups, and they had really rather sophisticated stone tools. And part of the reason I want to mention this too is that what we know now about Homo sapiens before in shall we say non-industrialized conditions is that they too lived in small groups, and that it wasn't really until the advent of agriculture where you see these much larger communities form, and that you begin to see organized religion in the sense of the idea of a divine lawgiver. Most hunter-gatherer groups have ideas about, for example, in the Inuit, there's the spirit of the narwhal and the spirit of the polar bear, but they had very sophisticated norms and ways for dealing with conflict, but they did not have the idea of a divine lawgiver. And I think this is important because we sometimes think that without that kind of religion, you wouldn't have morality. And that clearly seems to be false. So as I mentioned in talking about Ed Wilson, we know that there are lots and lots of species that are highly social, but the sociality that we see in mammals and in birds is really quite different. Now, the hypothesis to explain this has its locus in some really rather remarkable developments that took place about 200 million years ago. And that is that warm-blooded animals appeared on the planet. They were very small, and but they did have a really distinct advantage. They could forage at night when the other guys were lying around waiting for the sun to come up. And they could also live in colder climates. So it was a wonderful advantage. And it came, of course, with a cost. And the cost is that gram for gram, a warm-blooded animal has to eat 10 times as much as its cold-blooded cousin. So for Mother Nature, this is a problem. And one way to solve that problem is to wait until the genes provide for your knowing about how to succeed in your foraging. But that's kind of a long story. A faster way is to make brain wiring that learns and learns hugely. And that is really what happened in the case of mammals and birds. I should just, as an aside, say that in birds, the cortex doesn't look quite the same as it does in mammals, but the wiring ultimately is comparable. So what we see then is wiring that makes us really very flexible and very smart, that allows us to learn a lot about our environment and to make good plans, to get organized, to make tremendously powerful predictions about where the food is. And cortex is this sort of 6-layered structure. And the important thing, of course, about it is that when the infant is born, it's very immature. Now, this is an old slide, but it conveys very well the fact that neurons in newborns are in place, but they don't have much in the way of arborization in tree-like structure. They're very simple. However, very quickly, and you can see by 1 month, structure begins to come into place. And that's because the infant is learning. And connections are forming between neurons as a function of what it learns. It's estimated that in the newborn and the neonate, 1 month to 2 years, approximately 10 million connections, 10 million synapses per second come into existence. And so the strategy clearly for Mother Nature, if I may use poetic license here, the strategy of having wiring that allows for a tremendous amount of learning paid off quite handsomely. However, the strategy has a cost. And it works like the dickens as long as you have something that takes care of the neonate, because the neonate mammal is by and large helpless. And here you can see baby rats. They're blind, they're hairless, they need somebody to feed them and to keep them warm until they can manage on their own. And so Mother Nature had a problem, and the problem was, who's going to take care of these babies? Now, it's a problem because by and large, in reptiles or turtles, for example, the infant, in this case, a turtle, hops out of its egg, scuttles down to the water. The mother is long gone. She laid her eggs in the sand. She's nowhere to be found. So the wiring changed in the brain so that, yes, the offspring is going to have tremendous learning capacity. And yes, it's not going to be independent. It's going to be highly dependent. But the mother is going to be there when the baby is born, and the mother is going to have feelings of attachment to the baby. So how does that work exactly? Well, there's the sort of metaphorical way of explaining this, but there is a mechanistic story. The metaphorical way of explaining this is to say that just as we saw in the case of the deep values of self-survival, that wiring is modified in very specific ways so that the mother feels exactly the same urges, maybe even more powerfully, for the protection of her offspring. It's as though the domain, the ambit of me expands to include the baby. And that's not true. That turns out to be true of all mammals. So mechanistically, then, how is that achieved? Now, here the story is really complex because we know that there are many, many neurochemicals that play an important role. But the one that I'm going to focus on here, because it is very central, is oxytocin. So oxytocin is important in giving birth. It's involved in the contractions of the uterus that usher the baby out. It also is released in massive amounts into the brain of the mother. And as the baby is held and cuddled and there's skin-to-skin contact, it's released in massive amounts in the brain. Now, it's useful to know that the release of oxytocin, and here I'm showing you on the right-hand side, this green area is a region of the hypothalamus, which is a very ancient part of the brain. So this was what was changed to allow not only for oxytocin to be released into the body, but for it to be released into the brain itself. And on the other side, on the left-hand side, you can see this tiny region of hypothalamus from which oxytocin is released. And it goes into parts of the reward system, for example, into the nucleus accumbens, and into regions where the emotions are regulated. Now, there's another really important chemical for parenting that has been studied at Harvard by Catherine Dulac in her lab, and that's galanin. And I won't have time to talk about the role of galanin, but suffice it to say that other neurochemicals like galanin, like estrogen, like progesterone, are also very important. Now, you might say, okay, well, that's a good story about how you get an extension of the value of self to other, but how does that really play out with regard to morality? And here the story depends on a kind of surprising discovery by a field ethologist. And here is what they found. So there are many species of voles. Some are called montane voles, and they live in sort of rocky terrain and build their nests in burrows underneath rocks. Others are prairie voles, and they live in burrows, but search for food on the open prairie. So rather different environmental situations. And they're different behaviorally. So montane voles are kind of like what you would expect behaviorally. The males and the females meet, they mate, and then they go on in their separate ways. And the female has the babies and takes care of them, and the male looks for more action. The prairie voles are completely different. they meet, they mate, and now they're bonded forever. And what that means is that the male will help guard the nest. He helps take care of the babies. It doesn't mean that that's their only sexual contact, but it does mean that that's largely where the sexual contact takes place, is between the bonded pair. They are depressed if they're separated. And what that means is their cortisol levels, their stress hormones go up, and they don't feed properly and so forth. When the other mate is returned to the cage, they are joyful and they lick each other. Their stress hormones go down. And people say, well, what's the difference in the brain between montane voles and prairie voles? And the answer came out of Larry Young's lab at Emory University. And the answer is a little bit complicated, but not so very. And I'll talk you through it because it is the mechanism that's so important. So what you're looking at here are slices of brain from montane voles and from prairie voles. So just look at the top panel first. Now, what Larry Young's lab did was they used a stain that selectively picked up on receptors for oxytocin. Now, just as a side, you know that a neuron's not going to respond to a chemical unless that chemical can bind to a specially designed receptor on that neuron. So the receptors are the critical part of the story. And so here what we see is there are a lot of receptors, and NACC means nucleus accumbens, and that's an old structure that's a crucial part of the reward system. So when oxytocin binds to those receptors, it changes the behavior of the animal, and the animal also feels good because of the rewarding aspect of the activity. Now, in the montane vole in the same region, you see very little in the way of density of receptors for oxytocin. Vasopressin is very similar to oxytocin. Probably through much of history, there was only one peptide, and then it split into two, but they're very, very similar. And so they tested whether receptor density for vasopressin might also be different. And they found that, yes, indeed it is. And again, in the prairie vole, you see a difference here. That's part of the reward system. And we can talk about this later, but in montane voles, you see a high density of vasopressin in the lateral septum. And that actually has an effect of damping down attachment and bonding. So it depends on the wiring as well as on the neurochemical. So what's interesting about this then is that the difference in behavior between prairie voles and montane voles turns out to depend on receptor density of oxytocin. And of course, this was tested in all the ways you can imagine. You can take prairie voles, you can block their oxytocin receptors, and then they behave just like montane voles. So as I mentioned, that in the reward system, especially in the nucleus accumbens, there are pleasurable feelings associated with activity in those areas. And that's because in the nucleus accumbens and in the ventral tegmental area that are part of the reward system, there are endogenous opioids. They make you feel good when they are released. There are also endocannabinoids, and that's why we also feel good when they are released. All right. Now, once the discovery about the prairie voles was published in about 2004, many people got really, really interested in what precisely else oxytocin does and how it interacts with other neurochemicals in particular, and what the wiring is. One of the things that we do know is that, to a first approximation, that when oxytocin levels go up, stress hormones go down. And so part of the reason that a release of oxytocin feels good is that you relax. And that tends to increase the level of care and the level of trust between animals. So it's kind of a buffer against stress. And so when animals are, as for example, in this instance, they're a group of female chimpanzees that are sitting together and sharing food, this is exactly the kind of thing that raises oxytocin levels. I'll just go on to the next slide, because I think that in the last slide, the chimpanzees sharing food, it is a very interesting fact, because humans like to do this too. We love to share food. We have, our big Thanksgiving dinners and so on. And the theory is that food sharing makes oxytocin levels go up because it's co-opting mechanisms that support the mother-infant bond during the suckling stages. It turns out that there are some measures that show that doing good feels good. And I want to talk to you about one experiment here. So in this experiment, subjects volunteered and they were subjected to a painful stimulus, and that was a tourniquet. And as you know, when you have your blood pressure taken, it's after 10 or 15 seconds, it becomes kind of unpleasant. The subjects were had the tourniquet on and up to 180 seconds. So subjects were given an option when they did this experiment. They could either be paid and take that pay with them when they left, or they got paid and they could donate their payment to victims of an earthquake. And when they analyzed the data later, and the data have to do with the subjective rating that individuals felt during this long stretch of having a tourniquet on their arm, what they found was the subjective rating of donors was by and large much lower than the subjective rating of those who just took the money and planned to leave. It's a very interesting result because it has been mentioned, I think, as a suggestion in a number of instances that people do have feelings that are very positive when they engage in generous acts or brave acts and so forth. During this pandemic, we know, of course, that many people who are very isolated can become very anxious and depressed and so forth. And this really is related to a loss of social support. Brain oxytocin levels in animals are known to go down under these conditions. And it's a reasonable prediction that at least some of these feelings are associated with lowering of oxytocin levels. What does raise oxytocin levels in the brain? Well, interestingly enough, physical exercise does. Social interactions, we saw this in the case of food sharing. Social interactions of all kinds are important, but there's also data showing that social touch, just holding, touching, skin to skin, hugging, has an effect on raising oxytocin. If animals, this was done with mice, if animals, when they are neonates, don't receive the normal amount of cuddling for that species, I mean, as you know, rats and puppies and humans get a lot of cuddling. If they don't get that, what happens is that there's an increase in methylation of that part of the DNA that codes for oxytocin receptors. And hence there is a decrease in gene expression of oxytocin receptors decreased binding, and those individuals show rather different social behavior. In the case of female mice, for example, they're not very good mothers. And they don't show the normal social interactions that you would see in an animal that got lots of cuddling as a baby. Now, there are many, many species of mammals. Only about 8% show long-term pair bonding. Marmosets certainly do, but baboons and chimpanzees do not. So much will, of course, depend on and will reflect the kind of environmental conditions in which the animal evolved. And you can kind of see that in the case of the prairie voles, because living on the open prairie, the voles are subject to predation by birds like kestrels and hawks and so forth. And so if there's at least two mates that are working together, then that probably enhances the chance of having offspring that don't survive. Whereas in the case of the montane vole, the mom can go out and forage for food and leave the babies on their own, and they're pretty well protected. And the other thing that's so interesting too, I think, about the prairie voles is that the siblings of, if there is a clutch of babies and then they grow up and then another clutch comes, the siblings help take care of the babies. And there is also a kind of community amongst all of the prairie voles that live in a particular region. Wolves are, of course, super interesting to us because the dogs that we love so much descended from wolves. And wolves also are long-term pair bonders. And they show, as in prairie voles, when there is separation, they show very similar kinds of behavior. Now, in this illustrates something that's kind of special and wonderful about the evolution of very high degrees of sociality, and that is the animals can cooperate when they know and trust each other. And they also have a remarkable degree of understanding and being able to predict the movements and behaviors of other animals. So what you see here, of course, is the mother moose and the calf, and the wolves will bring her down. This one will rip the tendons in hind leg, she'll go down, and this one and maybe that one will come in and rip her throat out, and that will take care of business. This is very difficult to do, and it requires a tremendous amount of understanding of each other and how to pull it off. But of course, humans, often in much larger groups than the packs of wolves, also engage in a tremendous amount of cooperation. And the cooperation likewise involves strategies for reducing conflict, for ensuring that the outliers contribute in a way that they can, and so forth. So, just to go back to the question then about the link to morality, and I suppose I should have done this earlier, but let me do it now. By moral here, I'm going to mean something very simple. I mean just that I make a moral decision when I incur a cost to myself in order to benefit another. Now, philosophers are going to take that and, sort of complicate the heck out of it. But for the purposes of talking about social animals in general, that works quite well. So to kind of sum up where I've been going with this, it seems that sociality of the kind that involves oxytocin release begets other caring. And other caring, along with social learning, about the ways that things work in a group, about norms that obtain, then social learning begets social norms about right and wrong. They become habits that profoundly engage feelings of what's right and what's wrong. And of course, there's many ways of learning norms. Some of them involve imitation, but in all cases, the reward system is crucially involved. Now, the reward system, again, I mean, I think this is one of the things that surprise people. The degree to which the reward system is involved in learning very sophisticated things like norms. Because the reward system also is built out of ancient structures, the ventral tegmental area, the nucleus accumbens. Of course, it has projections to cortex, especially to prefrontal cortex. And for a long time, people thought that the reward system had to be really kind of stupid and that it really allowed for things like classical conditioning, but not much else. And one of the great revolutions, I think, in neuroscience in the last 2 decades has been the work that has shown how very complicated and very subtle are the things that the reward system learns. And we know, and Aristotle understood this very well, that we can have moral rules of thumb, but they aren't rules that apply always and every time, because situations change, situations are complicated. And the desire to have very simple moral rules is countered countered by what children see in their role models in a good and decent social life. Sometimes we have to tell a lie. It's the right thing to do. Not every day, as certain of our fellow Americans do, but from time to time. And growing up in a sort of religious context where we were given the 10 Commandments and told this, that tells you what's right and wrong, we all knew that you couldn't abide completely by the 10 Commandments and be a good person. So norms are extremely complex. They conflict with preferences. They conflict with each other. They vary across individuals and within an individual across time. But what kind of keeps it all together and keeps it all stable are the really important parts of sociality that we have by virtue of being social creatures. Now, in the last part of the talk, I just want to raise a question that at first I thought wasn't even worth pursuing. But the question is, does temperament play some role in political preferences or political ideologies? Now, the reason that this is a question that got asked depends on a group of political scientists, some of whom were at Nebraska. And John Hibbing and his colleagues in particular were interested in whether there were features of character, for example, that were themselves non-ideological, but played into ideological preferences in a given environmental context. And what they noticed was that at the behavioral level, when they did some of these experiments, that, for example, you show somebody pictures that are disgusting, and some people tend to focus on them much longer, and some people just have a look and then go on their way. And they noticed also that that tended to line up with explicit ideological preferences. So John Hibbing convinced Reed Montague to undertake an experiment in his lab, which asked the following question. For these non-ideological pictures, do we see any differences in brain responses between those who, shall we say, are strongly traditional and those who are strongly progressive? So the experiment, I think Reid Montague was actually very skeptical about doing it. And I was skeptical about the results at first. So the experiment involves 3 components. They put individuals, I think they had 83 subjects. They put them in the magnetic resonance scanner, and then they were shown pictures in the scanner, some of which were neutral or pleasant or threatening or disgusting. Once they finished that part, they were asked to evaluate how disgusting the pictures were. And finally, they were given surveys to determine something about their political ideology. And these surveys were tools that had long been developed by political scientists that tried not to sort of tap into current topics, but that would reach across generations to see what traditional values were held. So those are the 3 components, and I'll just show you again in this slide because it's important to see how this worked out. So there you would be lying in the scanner. For 4 seconds, you'd see a threatening picture. 4 seconds, you'd see a pleasant picture. Then you would see a blank screen with a square, and you had to press a button. And that was just to ensure that the subjects remained attentive during the whole deal. And then finally, somewhere in the list, you would be shown a disgusting picture. So what did the results show? Now, just before I give you the results, I want to show you what a non-ideological disgusting picture looks like. And this was an instance of some that were used. Others had to do with animal corpses lying in a field. Some had to do with human corpses lying in a field. So here are the results, and this is going to be a little bit complicated, but I'm going to simplify it because the basic story is quite simple. But let's just look at all this stuff on the bottom first. You see activity registered as yellow or red. Red is greater than yellow. And in all of these pictures of the brain, the activity levels are determined by seeing how much greater activity was to a disgusting picture than to a neutral picture. So you're really looking at data that are fundamentally comparative. Now, when they analyzed the data, they separated individuals into extremely conservative or extremely liberal as a function of their answers on the surveys. And what they found was we're going to look just now up at this top left-hand picture. What they found was that to a single picture, for example, worms in the mouth, there was a clear difference between those who came out as highly conservative on the assay versus those who came out as highly liberal on the assay. And you could look at that one scan of one individual, and with an accuracy of about 85%, you could predict where they were on the scale. So that was a surprise. And it was also a surprise because, well, 2 things. One was that although individually, all of these regions of the brain, for example, the thalamus or the amygdala and hippocampus, dorsolateral prefrontal, we know individually about what those areas do. But the idea that collectively they show a discriminative difference between responses to a disgusting picture left us all baffled. The other thing that was rather surprising, I think, was that individuals who showed a really high level of response to a disgusting picture, and comparably, or they showed a very low level, when they were given a rating of how disgusting these pictures were, there was no relationship between their brain response and their introspective report. So I might, for example, have found the picture of worms in the mouth really disgusting. I might give it a 9 out of 9. But my brain might not have shown that it cared very much. And that, I think, was also very surprising and very interesting. None of this, by the way, shows anything much about whether there are genes involved in this, or whether there are aspects of early childhood experience that are reflected, or adult experience that are reflected. The question of the causality for these differences is not shown, but that there are clear differences, but only to disgusting, not even to threatening, but only to disgusting, is a surprise. And this was the name of the paper, Non-Political Images Evoke Neural Predictors of Political Ideology. They gave it kind of a bland name, hoping that the more sensational newspapers would not pick it up. So I'm going to end really with just kind of a summary of what all of this data mean with regard to how we can think about morality. And one thing is that morality is not a module. It's not an encapsulated bit of brain organization, because moral decisions can be affected by all kinds of things. The norms that you've learned, by the current stress that you feel, by emotions that have been aroused as a result of various things that happened maybe in your history or that you expect to happen, and so forth. Aspects of risk aversion or temperament may also play in, and that may be why we see in the data from the Montague lab that there are actually differences in brain responses between highly liberal and highly conservative individuals. And finally, it's worth mentioning, because I love the work that's been done on animals from different species helping each other, that because sociality is pleasurable in mammals and birds, we may engage in behaviors that are largely unrelated to passing on our genes. And I discuss these issues in much greater detail in this book that came out a year and a half ago. And I would be happy now to take questions. And thank you for your attention.